pitfalls. Thank you very much, Serdar. I'm given the topic of talking about the pitfalls. As explained very well by Ali Saffet, these things should be done. International collaborations are a must.
Yaptığınız çalışmaların standartını yükseltmek için.
They will help you to raise the standards of your own studies and also contribute to the studies containing studies on humanity. In the last 10 years, collaborations impact has become so big that it is very visible. One of the most famous ones are the works conducted in the field of genetics in the last decade, in schizophrenia, in bipolar, and recently for depression, for ADHD. You know, the genetic studies like 30,000, 40,000 or 100,000 patients and healthy controls, genetic samples are collected and compared, and it won't be possible by a single group, single team to do it. It's only possible with collaborations. There is PCG study underway with contributions from different countries, and another example is the Consortium for Lithium Genetics, which is about the collaborations working in different countries who collected data, genetic data. And the results of this study were very good. Patients responding to and not responding to lithium were tested and published in New England Journal of Medicine and other journals. So it is clinically very significant. I hope that these genetic studies will become mature and it would be possible for us to identify the genetics of a bipolar patient whenever he enters into the room. We're not ready, but it goes to that direction. There are huge impacts of collaborations in brain imaging, in MRI studies, Enigma and Imagen. Enigma was mentioned by Ali Saffet. It's an international brain imaging consortium. Just like the case of genetics, the figures are low, the numbers are smaller, but it's growing. So they will make a comparison by collecting multi-site information. Imagen, both genetics and imaging data have been collected. It's a very good idea to participate in both of them as much as possible. But what are the pitfalls? I have divided them into two groups. The dangerous areas, the areas which we have to be careful about. The first one is about scientific and the second one is about logistical aspects. It's a little bit of an artificial differentiation. For scientific part, in translational research, what we are talking about, clinical trials, not about clinical trials, but about brain imaging, genetics, which are performed with patients, but higher biological aspects. Harmonization is the vital part. So different data from different corners of the world are collected together, and we should be sure that they can be compared. They are comparable. It is not easy to do that. Before the last 10 years, there was a statistical response, a classical response, collect the data, add a covariant, and for instance, data coming from the US should be numbered as 1, from Asia 3, Europe 2. So you give numbers to them, and if there is a difference, you can sort it out statistically. And for other statistical findings, it is not something very easy to do, both for imaging and both for genetics. The data structure can be so complex that adding a covariant cannot be possible to solve it. We should be more careful for imaging works. Brain images after derived from MRI device, they go through very different stages, they pass through very different stages, and we should be careful about that. And process data, if you are to collect it and compare it, it means that you lose the game, because in each and every region the process in US is different from the process in Europe. So if you are to harmonize MRI studies, the data coming right out of MRI device machine, it would be very good to collect it directly and try your best to find the differences and to correct the differences during processing. This problem is very well known in genetics. It's population stratification. So within the population, the stratification is made, and the first molecular genetics studies, when they were first started, you collect DNA data. However, you are in an hospital and in the emergency clinic, you get DNA data from many patients, and then you will collect from another segment and you will compare. However, due to a sociologic reason, patients coming to your emergency department may be different from the healthy subjects, healthy volunteers, because the patients are coming from the neighboring district of the hospital, and their genetic background may be different from the healthy volunteers. This is happening all the time. I am coming from the USA, from a multi-ethnic region. So we have to be very careful about unwanted consequences. When genetics works were started for the first time, there were bitter experiences. For instance, healthy subjects were all from Italy and the patients were from Puerto Rico. So they just make a comparison and they find genetic differences, but this is an ethnical difference. This is a background difference, but in modern genetics, the researchers have understood it better, and molecular genetics has advanced to a level that ethnical backgrounds, ethnical differences can be measured genetically. You look at 40 or 30 markers to identify the background of a person so that it would be possible to compare very different groups. And clinical population differences. It's just like ethnic stratification in genetics. A bipolar patient coming from a hospital and another patient going to another hospital may have different diagnoses on paper, may have the same diagnosis on the paper. However, there may be some very important and fundamental differences, education level, socioeconomic differences, the experiences, history of the patient, loneliness or abuse at the early ages. They may all have a huge impact. So clinical populations, patient populations should be comparable with one another, which is not an easy thing to do. And Professor Howe gave a very good example. In Indonesia, schizophrenia diagnosis, people living with that diagnosis within 10 years they are converted into bipolar, because the families, the physicians start asking some questions and then such transformation occurs. So if you had to do an imaging like 20 years ago in Indonesia, and if you are to do imaging now, there will be huge differences. We should be very careful. We have an endless list, an exhaustive list to give, but consortium should get together and the details of collaborations should be taught. Optimism is not something very easy, and we should be very careful about that to eliminate the systemic differences. We should either remove them physically or Detailed statistical data should be collected to ensure that different groups are equal to each other. For logistical aspects, these multi-site studies, their coordination is not easy. You have to exert a huge effort for recruitment and for assessment. Who will approach to the patients, who will be included in the study, and face-to-face assessments, interviews, and data, registering data to the database, who will be responsible for them. It may seem very easy, but I personally experienced this. Which resident or which assistant is there may change the situation. For instance, some assistants could interact differently with female patients or male patients. For instance, male patients are more involved, and for other assistants, female patients are more involved. This might be a sociologic relation, or this may stem from a sociologic relation. However, I should say that anybody who is there performing the study will have effects or impacts on the study. The best way, healthiest way to solve this is the joint education, training, and calibration, because in the multi-site studies, before the study, the contributors should get together, should spend some time together, should watch the interviews of patients, and they should evaluate MRI findings. This is very healthy, and it's been increasingly used day by day. So with such a joint education or training program, it has an impact, but this is not sufficient either, because at the beginning of the study everybody focuses on more overlapping factors, but within time culture of each group changes, evolves day by day, then they change their methods. That is why we need a calibration. We need to calibrate it depending on the study itself. Once in 6 months or once in a year, everyone should get together again, should share their experience, and they should compare their notes. Like, this is the situation in our study and this is the situation in your study. So this is something very healthy. And in MRI studies, there is something else which is difficult to prevent. Change of the equipment. MRI devices worth $1 million, and it's not easy for researchers to control it. Generally, hospital administration or university administration makes a decision to purchase a new one or to repair an older one or change the brand. So these are not under our control. However, if you start a multi-site work, for instance, a study performed in 8 or 10 different centers, an MRI study. In one of the centers, the university administration decided after 6 months to convert to Siemens from Philips. This is a huge problem. Change of device equipment is a huge problem. I mean, being optimistic is dangerous, and this is valid here as well. What are the differences between these two devices? And what about their systemic effects while collecting data? So our site, our centers, relations with other centers, and which devices are used in different centers, they should all be taken into consideration before these studies, and each and every individual should start using the old ones and new ones, and comparisons should be made. Sometimes they are creating matrix. So switching from Philips to Siemens, there is a mathematical configuration is needed, and after that time the data collected in Philips can be compared in another device. This should be taken on board, and this is a logistical factor. Another issue is that it may seem easy, but it's quite complicated. Who is going to evaluate data, and how will administration of or management of the study be performed, because everybody owns the data. However, nobody owns it alone. Everyone should get together, come together for their ownership. Our center is good at MRI or powerful at MRI. We will look at the thickness of cortex. That is what we think. And another center thinks that we are stronger in MRI and we will look at the thickness of cortex. However, if everything is based on hypothetical questions or assumptions, let's say, it becomes very difficult. So for multi-site collaborations, before initiation of the study, we should talk about how the analysis will progress. Sometimes tables are prepared like these, and these analysis will be done by this center and these should be done by another center. It's very good to do them in advance. If everybody agrees on that in advance, then everything becomes easier. It may be a tricky situation or difficult situation, but it's better to do it beforehand, because if you do it after the start of the work or study, things may become more complicated. And lastly, governance. Who will govern? Who will manage the process? Which committees will make the decision? So, like this is a multi-center involving 8 centers, and 7 centers got 5 patients per month, but the 8th center has 0 patients. So at one point, someone should speak with one of these centers and discuss the details, the reasons behind that lack of patients. They should be told in advance for the sake of the study, and if there are differences of opinion, you should decide beforehand to settle these disputes. There should be a regulation, something like a regulation that is prepared in advance to look at the details. So before the initiation of the study, by identifying the names like this and this will be in this committee, they will meet once a month and they will evaluate these issues. If a problem occurs, they will prepare a report. So a very detailed process is there, which is important for the sake of the study. As mentioned by Ali Saffet, for us it's very important, for those working in Turkey it's very important, and those who are to collaborate with us, they must be considered and taken into account in advance. So if possible, it is best to learn as much as possible and to discuss it with those who performed it previously, and try your best to get information as much as possible, and ensure that everything is written in a protocol format so that there won't be huge problems afterwards. Thank you very much.